So today we'll be discussing the process of translation. So unlike transcription and replication which occur within the nucleus, translation is a process which occurs in the cytoplasm. So it's a cytoplasmic process, and here the mRNA is translated from the 5' to the 3' end. And when the amino acid is being formed, it is formed from the amino terminal to the carboxy terminal. So first we have the first amino acid being formed, and then later on the rest of the amino acids formed, and finally we reach the carboxy terminal. Now let's see the phases involved in the process of translation. So first we have activation of the amino acid taking place, and the rest of the steps are similar to that of transcription. There's an initiation step, an elongation, termination, and post-translational processing. Now let's see each of these steps in detail. So first we have activation. So in activation, this is the basic step that is happening. Here what is happening is the amino acid needs to get activated. And for this, the enzyme that is helping us is amino-acyl tRNA synthetase. So this is the enzyme, amino-acyl tRNA synthetase. So here it's a two-step reaction. First what happens is the ATP gets activated. So the ATP is activated, and then this ATP will be converted to AMP. Now when this ATP is activated, the amino acid AMP enzyme complex will be formed, and this will help. So this activated amino acid will then bind to the tRNA, and then finally amino-acyl tRNA is being formed. So this is the activated form of the amino acid. Now let's see the initiation step. So for initiation to take place, first step is the recognition. So the first amino acid incorporated is the AUG codon or methionine in case of eukaryotes. So for eukaryotes, this AUG codon is methionine. And for prokaryotes, the same AUG codon stands for N-formyl methionine. Now, this is considered as a start codon, and this will be present after the marker sequence. So for the marker sequence, we have the Kozak sequence. This Kozak sequence is seen in mammalian cells, and it is Shine-Dalgarno sequence in the case of prokaryotic cells or bacterial cells. Now, for the further steps to happen, we require initiation factors or EIF. This E here stands for eukaryotic initiation. Now, next is a step where the formation of the 43S pre-initiation complex takes place. So we can understand this from the diagram. So here we have the met-tRNA, GTP, and the initiation factor 2. All of this will together form a complex, and then they will bind to the 40S ribosome with the help of initiation factors 3 and 1. Then they will together form the 43S pre-initiation complex. That is a pre-initiation complex. Now this will bind to the mRNA, and the 5' methylated cap of the mRNA is what helps in this binding process. And for this we need 1 ATP hydrolysis is taking place here, and factor 4 is enabling in this binding. So hence we have the 43S pre-initiation complex ready. Next is the formation of the ATS complex. So for this to take place, this whole complex, this 43S pre-initiation complex, is going to bind to the 60S subunit of the ribosome. This binding happens with the help of 1 GTP hydrolysis is taking place. That is why there's GDP and inorganic phosphate being formed. And also initiation factors 5 and 2 help in this process. And that is how finally the ATS complex is being formed. So here in this ribosome assembly, we can see two sites. There is a P site and an A site. The P site is a peptidyl site, and it carries a peptidyl tRNA. The A site is an amino-acyl site, which carries a new incoming tRNA. Along with that, there's also an E site. E site is the exit site. It carries the deacylated tRNA. Now moving on to the next step, it is a step of elongation. So in elongation, there are mainly three things happening. First is the binding of the amino-acyl tRNA to the A site. So talking about the binding of amino-acyl tRNA to the A site, so first we have the attachment of the new amino acid happening to the A site. So once this happens, GTP hydrolysis takes place, and with the help of initiation factors 1, EF1, the elongation factor 1, we have the binding of new amino acid happening to tRNA. Now, next step is the formation of the peptide bond. So for the peptide bond to be formed, the alpha amino group of the incoming amino acid in the A site will be forming the peptide bond with the carboxyl group of the peptidyl tRNA of the P site. So these two together will be forming a peptide bond, and for peptide bond formation we require an enzyme which is peptidyl transferase. This is the enzyme enabling the formation of the peptide bond. This enzyme is a ribozyme. It is a ribozyme. A ribozyme is basically an RNA acting as an enzyme. So that is how the peptide bond formation happens. And next is a translocation process. So now the tRNA fixed to the P site does not carry any amino acid, so it will be released. And then what happens is the whole ribosome moves over the mRNA distance, and now the peptidyl tRNA is translocated to the P site. It is moving towards the P site. That is how translocation to next codon is taking place. Now for this next step, we require a GTP hydrolysis taking place, and EF2 is responsible. So finally, a new amino acid will now attach to this A site. So here we have a new amino acid attaching to the A site. That's the translocation step. Now, before we discuss the energy requirements, let's see the last step, which is the termination step. So termination step, as the name suggests, will be the last step. So after successive additions of the amino acid, finally the ribosome will reach the terminator codon sequence. So the initiation sequence was AUG, methionine sequence. So for terminator, it can either be UAA, UAG, or UGA. These are the terminator codons. So it will reach one of these terminator codon sequences, and with the help of releasing factor 1, it will help in the process of termination. And here also there's GTP hydrolysis is taking place. So once that happens, we get free protein, free tRNA, and free mRNA, and the ATS ribosome will dissociate into its subunits, which is 60S and 40S. So that's the final step of termination. Now let's see the energy requirements here. So talking about the energy requirements, so for one peptide bond to be formed, we need 4 high energy phosphate bonds. Now where are these 4 high energy phosphate bonds being used? 2 are for the initial activation step, 1 for the step where elongation factor 1 acts, and another one for the step where elongation factor 2 acts. So those are the 4 high energy phosphate bonds we require. Along with that, we require 1 ATP for the initiation complex formation, 1 GTP is required for the ATS ribosome formation, and 1 GTP for the termination step. All of these are discussed in each individual steps. And very important point is that no energy is required for the actual peptide bond formation to take place. This is because the amino acids are already activated in the initial activation step that takes place. Hence, no energy is required for the formation of that particular peptide bond step. Now finally moving on to post-translational processing, there are various things happening here. Proteolytic cleavage can occur. An example for that is proinsulin being converted to insulin. Apart from that, there are various modifications of amino acids taking place. These include gamma carboxylation of glutamic acid residues, hydroxylation of proline and lysine and collagen, phosphorylation of OH groups of serine, threonine or tyrosine, co-translational glycosylation, disulfide bond formation, acetylation and methylation of kinin residues. These are some of the examples of various modifications of amino acids taking place in post-translational processing. That is all with translation.